So we went back into cells. This is one of the assays that we use to monitor this. So our protein, AGG32, is a mitochondrial protein. We can overexpress it. We can coat all of the mitochondria in our yeast cells, and they just kind of hang out. This is the inactive state. Our lysosomes are stained with this FM464. We can see in red here, it's a vacuole in yeast, but it's the same thing. Our green sits on mitochondria. It doesn't sit inside the vacuole. Once you now trigger mitochondrial degradation, you see a massive relocalization of this green signal to within the red. And this now tells us that our mitochondria have gone from just being in the cytosol, happy, doing their thing, to now getting into the vacuole and getting broken down. This is the normal way this works. What happens if we delete this domain? So we just strategically chopped out this domain, and we see the process is completely dead. Absolutely dead. Nothing at all. As if no mitochondria could be degraded no matter what we did. So we then took a sigh of relief and felt, okay, we can keep going. Let's see what we learn. So, and of course we can quantify this. So this is just the quantification. The black bars are the uninduced. The gray bars are the induced conditions. And what we see is when we delete that domain, we are at 100% baseline. Now, of course, we're messing around with the protein. We're changing it from its natural state. So whenever you do that, you could just mess it up. And it might be, the result that we're seeing here is really just because we completely destroyed the whole protein. Well, despite that, it still gets quite nicely onto mitochondria. So you can see the mitochondria are now stained in red, our protein stained in green, and you can actually see, because of the amazing resolution that we have on microscopes now, you can actually see that the protein sits on the outside of the mitochondria. So no matter what we did, here's the wild type, here's our mutant, which we played around with. This is still seems to be like the normal protein. It just can't degrade mitochondria. And of course, we want to make sure we have the same amount of that protein. So you can run it on an SCS page gel, do a Western blot. This band tells us how much protein we have based on the intensity of that band. I have to say, the first place I ever did a Western blot was here, and I probably did a lot of them. And yeah, it was, I don't know, again, just a really remarkable experience to be able to do that and to look back and realize I'm showing you a Western blot where I'm following the protocol that I used as an undergraduate here. So that was just kind of a weird thought that popped into my head as I was looking at that Western, but I made all of my students do Western blots exactly how Rachel Surmar told me to do them as an undergraduate. Pretty remarkable. So, okay, so we found this domain. It's completely required. If you get rid of it, everything stops. But let's confirm that this is really true. And so we used a second assay. And this assay is really fun. This is an enzymatic assay. So we have this little group blue protein. This is a phosphatase, which we can measure the activity of. This phosphatase only becomes active when it gets into the vacuole, lysosome and yeast. And so if we now target that, if we just lock it on the outside of mitochondria, the amount of this activity will be correlated to the amount that mitochondria have gotten into the vacuole and become degraded. So we can do the exact same experiment, and we see again with no induction, absolutely nothing. With the full-length protein, we get a really strong, robust degradation. And when we delete that domain, we see a complete loss of function. So this is one for the reviewers. So of course, whenever you submit any paper, the reviewers always have some really good suggestions and some really stressful suggestions. And this is one of those really sort of stressful suggestions. But thankfully, they bought this experiment. And so they were concerned that maybe there's some small region within the protein that correlated to this effect that we're seeing, and that the domain doesn't matter. It's just 5 amino acids somewhere within the domain that are really the important thing. And so, of course, we made all these smaller truncations and hoped 5 would be enough, and thankfully, they were okay with 5. So what we see is that absolutely no induction in any of these, which tells us, okay, this domain is really important and absolutely required. So at this point, we could think, well, what do we know about how AGG32 works? How it initiates mitochondrial degradation? And other labs had been studying this before we started, and so there was a little bit known. And what was known is that you have to phosphorylate 2 sites here at the N-terminus, and you have to cleave off the C-terminus by a protease to activate it. So we wanted to know, in the series of this, does our domain function before these or after these? So we can just chop out that domain again and see what happens. So as you cleave this part of the protein, you remove it, and now if we look at a Western blot, we see this band gets shifted and becomes a second band here. And as you let this go longer, you see the second band increase, and this is the result of removing this portion. If we delete our structured domain, we no longer see the appearance of this second band, suggesting that this domain functions pretty early in whatever this protein is actually doing. One of the things that this protein does is to recruit all the autophagy machinery, all the workhorse of generating this vesicle and isolating mitochondria. AGG32 is more just a switch, but a very complicated switch. Not just a simple on-off, but seemingly some sort of microcomputer built into a light switch, right? It's got all these different odd regulatory mechanisms, and how do those connect? But at the end, all it needs to do is get the autophagy machinery to mitochondria. So we check that. Is that lost? And what we see is it is. So this is a co-immunoprecipitation assay. Basically what we want to know is if we isolate this protein, do we see this protein come with it? So AGG32 is shown here. AGG11 is shown here. These are the controls just showing how much protein we have, and this is the experiment showing what we've isolated. So if we have no AGG32, we don't isolate anything, despite the fact that we have a lot of AGG11. If we don't have any AGG11, obviously we can't pull that down. We just see the 32. And then if we now express both, we see a strong interaction here, but a largely reduced one when we delete our structured domain. So at this point, we felt pretty confident that this domain that we identified was absolutely required, and that it functioned pretty early in whatever the protein is doing, and was pretty much required for everything else after. So I'm a structural biologist in training, and I told you at the onset that I really like looking at proteins and want to be able to determine the structures, and then I did not show you any protein structures at all. And unfortunately, that's sort of what we need to do to convince ourselves that what we're actually going to find will be informative. We need to be able to pair the structural biology with other techniques to make sense of what we're seeing. So at this point, we had a pretty decent idea of where we thought it might be functioning in this activation mechanism of this protein. So we said, can we determine the structure of this domain? So how do we go from this to this nice structure? So there's a number of ways to do this. The way that we did it, in this case, was NMR spectroscopy. So NMR spectroscopy is a fantastic technique. It's actually what I tried to do in graduate school, and then I got sidetracked to do X-ray crystallography, and then I continued to do X-ray crystallography, and then in starting my own lab, I got sidetracked to do NMR. So at this point, my department calls me a crystallographer, but I call myself a structural biologist. We do whatever we need to do to get job done. So NMR, this is a nice big magnet. You can put your sample in there, in this case a protein, and a lot of cool things happen. One of those cool things is peaks. So if any of you have looked at NMR spectra, you know that an NMR spectra is basically a series of peaks, and then those peaks correspond to our protons. Okay? So each proton gives us a single peak. Now in proteins, there's a lot of hydrogens. And so if you take a look at what a protein NMR spectra looks like, it doesn't look like a small molecule, it looks like a mess. Okay, so this is lysosome, 1,122 protons, which means you have 1,122 peaks. Your goal as an NMR spectroscopist is to figure out what each of these individual peaks are. Which proton in your protein do those correspond to? Unfortunately, that's pretty difficult to do with this spectra. Thankfully, other people developed much better techniques to do that. So, we can now separate our spectra with our 1,122 peaks into a second dimension by adding a nitrogen that now is active in our NMR experiment. And we can see this represents our hydrogen, this represents our nitrogen, and now we see resolved peaks that we can clearly identify. We can see, okay, here we have a single peak, here we have a single peak, so on. So, can we identify what each of these peaks are? What proton, nitrogen pairs do those correlate to? So, you can do that. There's a number of different experiments to relate those. That's a lecture for another day. But at the end of the day, we put HEG32 in the NMR spectrometer, the structured domain, and we got the HSQC, and it was amazing. I immediately put this on my wall and said, okay, we are going to get this structure. We're going to figure out what this thing looks like. So here's that HSQC. This is the hydrogen. This is the nitrogen. Each of these peaks represents a hydrogen-nitrogen pair in the protein, and thankfully, each amino acid has at least one, except for proline. They make NMR less fun, but they do. This one had a lot of prolines. So what we would expect to see in this is one peak for every amino acid in our protein, plus a peak for every side chain, like an asparagine, that has an NH pair in the side chain. So we can identify what each of these are, and we did that. Now, thankfully, we can separate even more into a third dimension, resolve our peaks even more. We can label our hydrogen, nitrogen, and carbon, and we can identify a frequency or a label for each of our atoms in our protein. So we can painstakingly go through and say, okay, the C alpha in valine 8 is going to be at this exact resonance frequency in our spectra. And when we do this for every single atom in our protein, we can then determine the structure. And the way that this works is through an experiment called a nosy. And nosies are really cool because now that you know exactly where each of your atoms are in these spectra, you can use another spectra that tells you how far apart they are. And basically, if those two atoms are within 5 angstroms, they will give a crossbeam like this. So we can take our simple polypeptide A, B, C, D, E, and if we look at this, we see C and E give a crossbeam, so they must be next to each other within 5 angstroms. We see D and B are also within 5 angstroms. So again, they must be close to each other. And we can use these distances to essentially fold our protein. We can take it completely splayed out. This is actually what the program does. And then it says, oh, this over here is close to this, so we better make them close to each other. Okay, what else can we restrict in space? And if you have enough of these distances, you can get actually your protein to fold in these programs. So here's a simple nosy spectra. You can see how much information is in this. All these peaks represent distances. At the end of the day, in this case, we had 2,422 distance constraints. So for our 140 amino acid protein, we have 2,000, a little over 2,000 distances that we're imparting. So we're basically taking a long string and then saying this is next to this is next to this, 2,422 times. And when you do that, you can get the atomic coordinates of your protein. This is the structure of AGG32, at least that fragment, and each of these represents a bond, and at the ends of each of these would be an atom. We can overlay on top of this all the distance constraints, and you can see how restricted this protein would be. It's not going to be able to fold into anything else, right? This is what it looks like, and those are all of the constraints, all of the NMR information that held it exactly like that. And so we definitively know that this is what the protein looks like. So we can represent that in a different way. Here's what that looks like. In NMR, we calculate a lot of structures to convince ourselves that we've done it correctly, right? So we calculate 20 structures, and if all 20 structures look the same, we feel pretty good. You can see that here. This bit at the end is disorder. So you can pretty nicely see how proteins have structured parts and parts which are very dynamic and can move around. So we didn't have distances in this region, and as a result, the protein doesn't really adopt any conformation. And if we go back, you can see that. There isn't any distances from here to here, but there's a lot of distances in that central region. So here's what that protein looks like. It's a pretty simple alpha-beta fold. It has a central beta strand surrounded by alpha helices. Here's what the secondary structure looks like written out. All right, what did we learn from this? Okay. We can return to our tool analogy and think, and I actually didn't know these were different. But again, when we have a protein structure, we want to know, well, what is this doing? What does this tell us? Why did we even waste our time solving the structure? We can return to this idea of these tools, right? So this and this are related, but different, as far as I've been told. And what we can see is there are features in this which are retained here, but this has some sort of extra handle that appears to have a locking mechanism, and so there's probably a similar thing that they can do, but maybe this one does it in a slightly different way. And the way we do this in proteins is to overlay them. We take one, put it on top of the other, see what's different. In the case of AGG32, we can do this. Now for the tool, we see, okay, well, this is smaller, this is bigger, this has this additional part. So we did this for AGG32. Here's what that looks like. Here's AGG32. Here's the most similar protein that we could find. And here's the overlay. So this wasn't too much of a stretch. This is what we actually do. We can see what's similar and what's different. Okay? So I'll skip a little bit of data, but just to say, this tells us that this domain could potentially act as a timer in which it gets phosphorylated and then it auto-dephosphorylates itself. In order to do that, it needs a number of residues. Those residues are these aspartic acids, this aspartic acid, and this serine. Well, these down here are classical, what are called receiver domains, which are most similar to this, and this is our protein. We don't have any of them. Okay? So this is this idea of what's similar and what's different. So we have no ability to act as a timer, but we seem to maybe be doing something similar. So with that, we did some other additional experiments to show that, yes, we could make mutations and mess up the function, and we could refine our hypothesis. And at the end, what we're left with is a new idea about how this protein works. So it sits in mitochondria. Then something, working on identifying what that is, but I don't know today, activates, we believe, a protein recruitment to this domain that we saw the structure of. This causes some structural change to lead to this proteolytic event, and then that creates another change, which leads to the recruitment of the autophagy machinery to mitochondria. We think that's probably how it's working. Turns out it's a lot more complicated. I will skip all that data. I left it in here just in case I talked really fast, but I did. So there's another exciting part. It's on the way. Look for the paper, 2019 maybe. But I do want to put in a short plug for Dartmouth. So we have 2 graduate programs. If you're interested in graduate school, we have a molecular and cell biology graduate program at the med school, which is across the street. We have a graduate program in chemistry. So I'm actually in the chemistry department. I teach biochemistry, general chemistry, all sorts of great chemical biology things. But these 2 programs are not so different. So I accept students from either program. Most of the biochemists do. It's a really great community. It's very collegial in a way that I've never experienced really anywhere else. People read my grants, actually, like carefully read them, carefully read other people's papers, give you feedback. It's really fantastic. And if you like being out in the woods, well, Dartmouth is out in the woods. I moved from DC, so I'm still trying to figure out how to be outdoorsy. I see sometimes other professors out hiking with their kids, and I'm somewhere on the side of the trail catching my breath. With that, I just want to point out the people that did the work. I've been really fortunate in starting my lab to work with a great group of people. So Sherry, who's a graduate student shown here, she did the bulk of the structural biology, and our postdoc, Sarah Katzenell, right there, she did the bulk of the cell biology. I've been fortunate to have a lot of funding. I got an initial grant from the institution to support this work. I got pulled into a program grant from the NIH, and I now have my own independent funding through the NIH as well. So thank you all for coming out on the Friday afternoon.